Hello, welcome friend. Welcome to our channel Quality Guru. Friend, today our topic is clean room classification pharmaceutical industry. Pharmaceutical clean room classification involved two main standards. The ISO 146441 standard, which defines cleanness classes from ISO 1 to ISO 9 based on airborne particle count per cubic meter, and the second is the UGMP Annex 1 guideline, which categorize rooms into grade A to through grade D for different level of contamination control in sterile product manufacturing. An ISO 5 clean room is a common requirement for sterile operation, while the GMP grade provide a framework for managing risk during the manufacturing process, ensuring patient safety and product efficacy by minimizing airborne particulates. Clean room classification provides a standardized way to categorize control environment based on their label of air cleanness, especially measuring the concentration of airborne particles. The primary objective of the classification is to measure maximum number of particles of specific size allowed per cubic meter of air. For example, when we measure particles that are 0.5 micrometers or larger, that is about 1 upon 200 the width of a human hair, we can determine if a clean room meets particular classification requirement. ISO 146441 classification basis. This international standard classifies clean room by the maximum number of airborne particles allowed in a given volume of air, that is per cubic meter, as a specific size. Classes. The scale ranges from ISO 1, that is the cleanest, to ISO 9, that is the least clean. Pharma application. An ISO 5 clean room is typically required for sterile processes and aseptic filling, as it significantly minimizes the risk of particulate contamination in pharmaceutical product. Federal Standard 209E. The Federal Standard 209E has been officially discontinued since 2001. However, it is still historically significant and is still referred in many existing facilities. That is why it is still worth explaining. It was developed in the year of 1963 by US General Service Administration and has since served as a primary clean room standard until the adoption of ISO 14644. ISO 14644, the global benchmark. The International Organization for Standardization, that is ISO, developed ISO 14644 as a comprehensive framework for clean room classification and testing. This standard can be divided into 6 primary parts. They are as follow: classification system overview, technical requirement and measurement, that is particle size consideration, testing and verification method, that is airborne particle count testing, classification states, documentation and compliance requirement, impact on industry practices. Classification system overview. The ISO classification defines 9 classes ranging from ISO class 1, this is most stringent, to ISO class 9, that is least stringent. Each classes specify the maximum allowed concentration of particles at different size measured in particles per cubic meter. The standard uses a logarithm scale where each subsequent classes allows approximately 10 times more particles than the previous one. Technical requirement and measurement, that is the particle size consideration. The standard measure particle in 6 size ranges: 0.1 micrometer, 0.2 micrometer, 0.3 micrometer, 0.5 micrometer, 1.0 micrometer and 5.0 micrometer. Each size category has specific concentration limit depending on the ISO classes. For example, in ISO class 5, the maximum allowable concentration for particles is greater than equal to 0.5 micrometer is 3520 particles per cubic meter, while for particles is greater than equal to 5.0 micrometer, it is only 29 particle per cubic meter. Testing and verification method: airborne particle counting testing. The standard mandate specific testing procedure using calibrated particle counter. These instruments must meet the requirements outlined in ISO 215014, ensuring measurement accuracy and consistency. Testing typically involves systematic sampling across designated locations, statistical analysis of particle count data, documentation of environment conditions during testing, regular calibration of measurement equipment. Classification state. The ISO 14644 recognizes three distinct operational states: as built, as built, at rest, and operational. With respect to as built, complete and ready for operation but without equipment or personnel. At rest, complete and ready for operation along with all services functioning and equipment installed but without personnel. Operational, normal operation with all services functioning and personnel present. Documentation compliance requirement. Maintaining ISO 14644 compliance required extensive documentation. This including but not limited to initial classification, testing result, periodic monitoring data, equipment calibration record, environmental parameter logs, personal training record, standard operating procedure, that is SOPs, contamination control strategy. UGMP Annex 1 classification grade A, B, C, D. Factor affecting clean room. There are 5 other factors that also influences the classification. Number 1, air filtration system. Effective air filtration is essential for maintaining clean room standard. Clean room typically utilize HEPA filters or ULFA filters to remove particulates from the air. The efficiency and the type of filtration system directly impact the clean room ability to meet its designated classification. Air changes per hour. ACPH. The frequency of air changes is another critical factor in clean room design. Higher ACPH rates are necessary for cleaner classification. For example, an ISO class 7 clean room required between 60 to 19 ACPH to maintain its standard. This assures that contaminants are continuously removed and replaced with the filtered air. Number 3, environmental control parameter. Temperature, humidity, and airflow pattern must be strictly controlled within a clean room. Variation in these parameters can lead to increased contamination level and affect the performance of sensitive processes. For instance, maintaining specific temperature and humidity level can help prevent condensation and microbial growth. Number 4, design and construction material. The material used in the constructing in a clean room, such as wall, ceiling, floor, and fixtures, play a significant role in classification. Smooth surfaces that are easy to clean reduce particle accumulation and contamination risk. Additionally, the overall design must facilitate proper airflow and minimize dead zone where particles can settle. Number 5, intended uses and process requirement. The specific application for which a clean room is designed will dictate its classification requirement. Processes that are more sensitive to contamination will necessitate higher cleanliness standards, influencing both the design and the operational protocol within the clean room. Why is classification important? This classification system helps ensure patient safety by controlling contamination risk at every stage of the sterile drug production. Patient safety. The primary goal is to protect patient from the contaminated medication. Product efficacy. Clean room classification ensures the medicines are effective and free from harmful contaminants. Regulatory compliance. Regulatory bodies like the FDA mandate these standards to ensure the safety and the quality of pharmaceutical product. UGMP Annex 1 classification grade A, B, C, and D. The EU Guide to Good Manufacturing Practices provides this classification for sterile medicinal product manufacturing. Grade A, the most stringent label equivalent to ISO 5 clean room used for high risk operation like sterile filling lines. Grade B, a background environment for grade A area also requiring very high cleanliness level. Grade C and D, less critical area with lower but still defined cleanness requirement than grade A and grade B. Key aspect of clean room. Key aspect of pharmaceutical clean room classification are the adherence to the ISO 146441 standard, which classify room by the number of airborne particles, that is classified by size in micrometers, and the control environment parameter like airflow, temperature, humidity and pressure. Other crucial factors include smooth, non-shading surface, effective HEPA filtration, strict personal protocols with proper gowning and the entry procedure, and the continuous monitoring and the validation to ensure required cleanness level is maintained. Classification by particle count and ISO standard. Clean rooms are classified the maximum allowable concentration of airborne particle per unit volume of air, ISO 146441. This international organization for standardization standard is the basis for classification ranging from ISO 1, that is most stringent, to ISO 9, that is least stringent particle measurement. The classification is based on the number and the size of the particle, typically measuring particle from 0.1 micrometer to over 5 micrometer using a cubic meter of air as the reference. Key design and environmental control. Air filtration. HEPA, high efficiency particulate air filters, are essential, capturing at least 99.97% of particles 0.3 microns in diameter to remove contaminants. Control airflow. Unidirectional laminar airflow, often achieved with ceiling mounted filter, help prevent cross contamination and particle deposition. Pressure differential. Maintaining higher pressure in cleaner zone than a surrounding area prevent contaminated airflow from migrating into cleaner area. Temperature and humidity. HVAC system regulate temperature, that is 18 to 24 degrees Celsius, and humidity, that is 45 to 60%, to ensure stable working conditions and minimizing particle generation. Thank you. Thanks a lot, and keep watching our channel. Material, like, comment and subscribe please, more and more. Thank you.